Hi, I'm Rico Amato. I'm a first year PhD student in Dr. Forney's lab at the University at Albany. My project is about the development and use of a mouse model to lineage trace the elusive terminal nerve. During embryonic development, the olfactory placode gives rise to a group of neurons called gonadotropin releasing hormone neurons, or GnRH1 neurons, in the area that will become the vomeronasal organ, or the VNO. The GnRH1 neurons, along with other migratory cells and neurons, form the migratory mass to migrate from the nose to the brain. One of these neuronal types within the migratory mass is thought to be the terminal nerve. The GnRH1 neurons have been found to migrate along the axons of the terminal nerve to invade the brain. The terminal nerve is also the only known nerve to project from the nose to the basal forebrain. To demonstrate that the terminal nerve is a separate set of migrating neurons from other olfactory neurons, this staining was done. In this immunostaining, OMP, which is olfactory marker protein, is labeling olfactory neurons, and in red is TAG1, a gene that is known to be a marker for neurons actively projecting their axons to their final targets. The terminal nerve is seen to be a distinct structure based on the staining, since no colocalization between the two markers is seen in the axons going towards the hypothalamus. So this project's main questions are where exactly a terminal nerve is, and are there other neurons that migrate from the nose to the brain? To label the terminal nerve, first we have to understand that there are two different stages of neurogenesis in the VNO. An early stage that produces migratory neurons, and a later stage that produces the resident sensory neurons. In this immunostaining at E11.5, when GnRH1 neurons begin to migrate, you can see in green is GnRH1, and in red is TAG1. Migration is occurring, and that a lot of the migratory mass, including the GnRH1 neurons and the putative terminal nerve, is expressing TAG1, while only a few neurons express TAG1 from the olfactory epithelium. At E15.5, the terminal nerve can be seen invading the basal forebrain. After this stage, however, TAG1 expression starts to go down, making the terminal nerve harder to identify than the earlier stage. By immunostaining at these time points, we have revealed that TAG1 is transiently expressed by both the GnRH1 neurons and the terminal nerve and can be a reliable genetic entry point to trace these neurons. To take advantage of this, we have generated our own tamoxifen-inducible Cree mouse line. The contactin-2, or CNTN2, or TAG1 mouse line, contains a Cree ERT2 construct shown here with the autocleaving protein P2A. When translated, P2A can cleave itself to separate both the Cree ERT2 and TAG1 proteins, yielding an efficient method to obtain separate and equivalent amounts of both proteins, meaning we generate a knock-in mouse model that does not affect the native function of the TAG1 protein. This line is currently viable and is continuing to propagate. Some preliminary data we have of this tracing is here in this figure. We've bred the TAG1 Cree RT2 mouse with a Rosa TD tomato mouse and injected tamoxifen at E11 and E12 to trace migratory neurons. In green is GnRH1 immunostaining, and in red is TAG1 lineage tracing. Looking at the VNO, we can see that very few neurons within the VNO itself are traced, and we can also see several of GnRH1 neurons are traced. And interestingly, we also trace several other migratory neurons that are not GnRH1 neurons. Looking at the forebrain junction, we can see that traced GnRH1 neurons invading the brain alongside other traced migratory neurons that are not expressing GnRH1 and only express TAG1. This tells us that this mouse model effectively highlights migratory neurons coming from the nose that are not GnRH1 neurons. To summarize, both GnRH1 neurons and neurons of the terminal nerve transiently express TAG1 protein, making them hard to track in later stages of life. To get around this, our mouse line can be used to effectively track TAG1 expressing neurons that can be timed with tamoxifen injections. And for future use of this mouse model, we plan to pair BRDU injections during the early neurogenic phase of the VNO with single tamoxifen injections at later stages, collecting different animals at the same time age of E14. This will allow us for the first time to birth date and trace the terminal nerve and characterize the other unknown neuronal cell types that form the migratory mass. Thank you for listening.